Our next speaker is Shamus. Shamus is a graduate student in my lab and is part of the Bioinformatics interdepartmental PhD program here at UCLA. Actually started in my lab when we were still at the University of Kansas, and moved here to LA with us when I moved my lab here. And he's really driven a lot of extremely interesting work in my lab and helped us sort of establish a new area of research. The work that he's going to talk about today has to do, and I think matches up very nicely with some of the things that Olga just talked about. And it has to do really with how do we analyze and understand this sort of rapidly exploding body of single-cell RNA sequencing data. Without any further ado, I'll let him take it away.
Hi, thanks for having me. So what I'm going to be talking about today is dimensionality reduction in single-cell RNA sequencing, and how, if you think critically about some of these dimensionality reduction workflows, you can find things in the data that you would miss if you were to just use a textbook approach. The system that we're studying is small cell lung cancer. Now, this is the type of lung cancer that you don't want to get. Usually by the time it's diagnosed, it's already began to metastasize, and that means that surgery, in addition to the tumor being diffused throughout the lungs, it means that surgery is not usually a viable option for patients, and that also makes obtaining tumor samples difficult because nobody wants to have their lungs cut open if it's not going to help them get better. So because it's such a serious illness and because it's been so difficult to come up with new treatments, the National Cancer Institute has designated small cell lung cancer as a recalcitrant cancer. And that's part of the genesis of our project, which is a collaboration with our colleagues at Vanderbilt University in the Caranta and Lopez labs. And what we're doing is we're looking at single cell RNA sequencing of small cell lung cancer cell lines. And we want to see if we can make some progress in the understanding and treatment of this disease. Now, we hear heterogeneity a lot, and the word can have different meanings depending on the context. And so in the context of this talk, what I mean by heterogeneity is you have a tumor, you give it some type of treatment. A lot of the cells die, but some of them persist and proliferate and eventually metastasize and kill the patient. So it would behoove us to know what the difference is between those cells that die and those cells that persist. There's also heterogeneity in terms of patients' response to treatment, but we really can't speak to that with this work because we're looking at cell lines and not actual tumor samples from patients. Now, it's been known since 1985 that there is heterogeneity in small cell lung cancer. At that time, cell lines were established and they were treated with chemical agents, and it was clear that there was separatrix between the samples. There wasn't a lot of progress made until the development of RNA sequencing, and at that point, we could start to see which genes were more highly expressed in one subtype versus another. Now, eventually, it was found that there might be a third group, and that third group was eventually subdivided into two more categories. In 2018, our collaborators in the Caranta lab developed a Boolean network-based approach, and they decided that the original subtype, the classic subtype, ASCL1 high subtype, should be further subdivided into two categories. Now, this review article that I'm taking this from illustrates that they think that, but then they decided they decided to propose a nomenclature that ignored our collaborators' work. And I don't know what's up with that. I'm staying out of that. But so this is in the field, there's some debate over the different types, and this is a proposed nomenclature. And as you can see, there are therapies that work better based on sequencing results. And so what we'd like to do with single cell RNA sequencing is we'd like to understand further what's going on here and maybe better characterize these different types of small cell lung cancer. So our outstanding questions here are, what is the best way to categorize subtypes of small cell lung cancer? And as you can see from the previous slide, there are different schools of thought. And really what we've seen in our work is that all the schools of thought are wrong. And what we should really be asking is, what are the functional roles of different cell populations to the extent that they can be defined? And then another question that we have is, what roles do Notch and Wnt signaling play in intratumoral heterogeneity? We know that there are often inactivating mutations in the Notch signaling family in patients that have small cell lung cancer. And we also know that Wnt signaling is often upregulated in the ASCL1 subtype of small cell lung cancer. And so if we were detectives working a case, these would kind of be our leads that we would want to look at. The experimental technique that is the genesis of our data is single cell RNA sequencing. And I know most of you already know how this works, but basically there's a barcode and it's coupled with a single cell in a droplet of oil. Sometimes two cells get in there, but there are methods to filter those doublets out. The cell is lysed. So single cell RNA sequencing is a destructive assay, which is not ideal because you destroy the thing you're studying and you can't go back and take a second look at it. But it does give us a clear picture of all of the messenger RNA that are in the cell at that time, and that gives us a picture of the gene expression at the time of lysis. And so what we end up with is a huge data matrix with 20,000 to 40,000 dimensions, each corresponding to a gene, and rows corresponding to a cell. And a lot of these dimensionality reduction workflows were meant to sort of condense this giant data matrix into a more manageable format. So what's typically done is the first step of dimensionality reduction is dimensionality reduction by subsetting. What the most popular approach is to take the genes that vary the most from cell to cell and just look at those. And there's an arbitrary cutoff of how many genes you would select. It's usually between 1,000 and 5,000. And then what's typically done after that is principal component analysis is used to further reduce the dimensionality of the data set to between 10 and 100 dimensions. Some groups do then analyze the data at that point, and then some groups will reduce the data even further using a nonlinear dimensionality reduction algorithm like t-SNE or UMAP, and then they can also visualize the data once it's in 2 or 3 dimensions. So what's typically done is that clustering and cell type identification is done either in the PCA space or in the t-SNE or UMAP space. And then visualization is typically done with t-SNE or UMAP in 2 or 3 dimensions. Now, I want to talk about linear versus nonlinear dimensionality reduction because I think it's important to understand. Linear dimensionality reduction, like PCA, it cannot bend or warp the data at all. It can only rotate it or translate it and find the way that the projection into 2 dimensions that best preserves the structure of the data. Nonlinear dimensionality reduction is attractive, even though it takes longer to compute, because it can sort of, as you can see here with the S curve, it can sort of unfold the data and we can get a clearer picture of the manifold than we would with linear techniques. And so nonlinear dimensionality reduction is attractive, but t-SNE takes a lot longer than PCA. And it has other drawbacks, which we'll see in a moment. Now, this is an example of dimensionality reduction that we're all familiar with. The problem is, how do we represent the Earth, which is 3-dimensional object, in 2 dimensions on a map? So we have some choices. We can explode, we can sort of stretch Greenland and we can stretch Antarctica so they're much bigger than they are in 3 dimensions. Or we can make cuts so that Greenland is cut into 2 pieces and Antarctica is cut into 4 pieces. Either way, we're introducing distortion. The 2-dimensional representation is not a faithful representation of the 3-dimensional object. There's always going to be distortion no matter what strategy you use. And so the way that this project got started is we wondered, is there a way to quantify this distortion? Can we somehow put a number to how distorted our dimensionality reduction is? And the way that we do that is something called average Jaccard distance that we developed in our lab. And basically what we do is we take a point in the original data and we look in the same point in the embedding, the lower dimensional representation, and we look at the nearest neighbors. We take the 100 nearest neighbors or 20 nearest neighbors or however many in the high dimensional space, and we take the same number of nearest neighbors in the low dimensional space. And so we have 2 sets that are the same size, and then we compare the intersection of those sets. What you see here is the red dots are the nearest neighbors in the high dimensional space. The blue are the nearest neighbors in the low dimensional space. And then the pink is are points that are nearest neighbors in both the high dimensional space and the low dimensional space. The way we quantify that is with something called the Jaccard distance. And it's just a measure of similarity between sets. A Jaccard distance of 0 means that there's no difference between the sets. The neighborhood in the high dimensional space and the low dimensional space are exactly the same. A Jaccard distance of 1 would mean that the nearest neighbors are entirely different. There's a whole new set of neighbors in the low dimensional space. Now, that's for a single point. If we average that over the entire population of cells, we get the average Jaccard distance. And that is a measure of distortion brought on by dimensionality reduction. Now, in our method, we don't discriminate between the second nearest neighbor and the 10th nearest neighbor. And we would like the second nearest neighbor to be closer than the 10th nearest neighbor, but we haven't figured out how to take the rank of nearest neighbors into account just yet. That's something we're still working on. So just to remind you, no distortion, we get an average Jaccard distance of 0, and total distortion is an average Jaccard distance of 1. So if we have our simulated data sets here, our S curve and our sphere, and we use t-SNE to represent them in 2-dimensional spaces, you can see that the S curve is a little less distorted. I mean, it's stretched out a little and there's some gaps in it, but the red is next to the orange, is next to the yellow, whereas in the bottom 2D representation of the sphere, you see that things are a little more scrambled. And so what we would expect if our method works is that the average Jaccard distance of the top representation would be a little lower than the average Jaccard distance of the bottom. And that's indeed what we find. And we tested this not just on this data set, but we had a lot of simulated data sets that we've tested this idea on. And we've also done a lot of testing on actual single-cell RNA sequencing data sets. And so if you'll look again at this dimensionality reduction workflow, you can see that we're going from 20,000 to 40,000 dimensions all the way down to 2 or 3. And so if there's distortion going from 3 dimensions to 2 dimensions, one would imagine that there's a lot more distortion going from 20,000 dimensions to 2 dimensions. So what we have here, this is from the Giuliano lab at UC Davis, and they study Hydra, and this is from a 2019 paper, a really good paper from Science. And this is one of the data sets that we tested our technique on. And what we found is that this figure has an average Jaccard distance of 0.85. And so what that means is that 85% of the nearest neighbors in the 2-dimensional representation are different than in the raw data. And that's a big problem because most of the algorithms that are used to analyze this data depend on a nearest neighbor search as their first step. So if 85% of your nearest neighbors are different, your analysis is being done on 85% just artifacts of dimensionality reduction. So that's a problem. And we also looked at this figure shows if we look at the entire data set and reduce its dimensionality, we get really high distortion. If we look at a single cluster, then we get slightly lower distortion, but it's still very high. Even in the best case scenario, it's above 60%. So we would like to develop better dimensionality reduction techniques that can give us a better solution to this problem. And that's an active area of research in our lab. But for our small cell lung cancer study, in the meantime, we wanted to find if we did our clustering on the raw data and then just use dimensionality reduction for visualization, if we wanted to see if maybe we would see something that we wouldn't otherwise. And so that's what we did. And right here, we have UMAP representations. This is clustering done with the Louvain algorithm on the raw data, and then it's visualized in 2 dimensions with UMAP. And now, if you remember the classification scheme, the first thing that we wanted to look at is whether our cell lines, our 8 cell lines that we got from our colleagues at Vanderbilt, if they corresponded to this classification that was based on bulk RNA sequencing. And so we looked at the distributions of the marker genes, and what we found was very surprising. We found that the classification scheme really doesn't hold at all. MYC and MYCL are supposed to be, if one's down, the other should be up, but we find that that's often not the case. The marker genes are supposed to be, if, for example, ASCL1 is high, NEUROD1 should be low, but we find that that's often not the case. And we also find, and in addition, that a lot of these distributions are multimodal, that what we're looking at here is really a complex ecosystem, and that perhaps dividing it into bins or categories isn't the best way to conceptualize this system. So the next thing that we looked at was whether our clustering that we got with the Louvain algorithm in the raw space corresponded to these marker genes. And we found again that these marker genes that have been previously studied were not a great way to differentiate our clusters. And at this point, we didn't know if, well, maybe our clusters just don't mean anything. But we wanted to find out for sure. And so we devised another test. Now this test, we used PCA on the data, and we embedded it in every dimension from 1 to 1,500. The reason that we didn't go higher than that is because PCA needs a certain number of samples that's greater than the number of dimensions in order to be stable. And above 1,500, we didn't have enough samples. And so what we did was we evaluated the average Jaccard distance for every single one of these embeddings. And then we compared each cluster, and we took, as a control, we took a random subsample of the data that was the same size as each cluster, and that's the dotted line. So the dotted line is a random subsample, and the colored line is each cluster. What we found was that the topology of these clusters varied widely. And if our clusters didn't mean anything, we would expect all of these lines just to match up with the random subsample. But clearly there's a difference in the topology of this cluster 3 here. It has a much lower average Jaccard distance, which means that the topology of this cluster is easier to represent in lower dimensions. Now, the next thing we looked at, we wanted to see what genes were highly expressed in our red cluster, cluster 3, as opposed to the other clusters. And for the remainder of this talk, I'm going to use the cell line DMS53, but the other 7 cell lines, we got similar results. So this is just an illustrative example. We found that there were several genes that were highly expressed in this cluster. And then when we looked them up in databases, we realized that a lot of these genes had not been studied at all. And so nearly nothing is known about this. And that's very exciting because these present what might be targets for new treatments. And so we don't specialize in the study of individual genes in our lab, but we have this list of genes that we think might be interesting targets. And we're hoping that when we publish this work that somebody will take this list and kind of run with it. The next thing we looked at was the average number of counts per gene. we wanted to see if maybe there was some numerical reason that our clusters, and what we found was that the average count per gene was higher in our cluster 3 versus the other clusters. And so that's interesting because it's known that stem cells have a higher average number of counts when they're sequenced. And so another thing we know about stem cells is that the number of genes expressed per cell is higher in stem cells. And so when we tested that, we found that our special cluster also had a higher number of genes expressed per cell. Further, when we looked at notch signaling, which was one of the things that we thought was interesting, or could be interesting based on previous work, we found that that was very differentially expressed. It was inactivating in our special cluster. And that's very interesting because it's been found that patients have inactivating mutations in small cell lung cancer. And so this, we might be looking at getting closer to the source of the disease here. And then, so one of the, our techs in our lab, Tim Hamilton, who's now a grad student in our lab, he found this algorithm that was developed at Stanford called Cytotrace. And Cytotrace is a mathematical model that's been trained, it's a supervised classification algorithm, and it's been trained on stem cells and non-stem cells. And so basically this model, once it's trained and you give it any cell, it can give you a probability between 0 and 1 that that cell is in fact a stem cell. So when we fed our data into the Cytotrace algorithm, we found that it gave a very high probability that our special cluster of cancer cells were stem cells. And so all of these things taken together, we find that this special cluster, and by the way, we would have totally missed this cluster if we had used the textbook workflow where we do HVGs, or sorry, highly varying genes, and then PCA, and then t-SNE. We wouldn't have seen this at all. But when we do see it, we find a cluster of cells that has several properties that are different than the rest of the population and also correspond to stem-like cells. And so I think that's very exciting because those might be potential targets for treatment, and we might finally make some headway in treating the disease. So our next steps, we'd like to treat the cell lines with therapeutic agents and sequence the resulting populations. We'd like to see if we get the same population dynamics in cell lines that have been treated with chemotherapeutic agents as we do with the untreated cell lines. We'd also like to sequence patient-derived tumors. They are difficult to get, but it is possible. And that would tell us, when we're studying things in cell lines, we don't ever know for sure if the biology is the same as it is in the human body. Or actually, that's not true. We do know for sure that it's different. So we'd like to see if the phenomena that we're seeing is also observable in patient-derived tumors. We'd also like to isolate single cells and then regrow the population from single cells and see if these different types of cells that we have are capable of recreating the entire population from a single lineage, or if only some of them have that potentiality. So I'd like to thank everybody in the Deeds Lab for all their support and feedback. Timothy Hamilton and Samuel Aragones especially, they worked very closely with me on this project and put a lot of work into it. So I want to definitely acknowledge their efforts. I'd like to acknowledge my committee members and also our collaborators at Vanderbilt University who actually did the experiments that sent us the data. So with that, I'll conclude, and I'd love to answer any questions that you have.